This is the third and final video in the introductory lecture on plant pathogens. It includes Basidiomycetes, oomycetes, and Plasmodiophoromycetes, focusing on families and genera that are important in fruit or vegetable crop production. Basidiomycetes are an important group of true fungi that contain numerous economically important plant pathogens. Hyphae are septate, but Basidiomycetes have evolved a mechanism that allows the exchange of nuclei upon mating of hyphae of different mating types. The arrow in the micrograph points to a clamp connection, and the diagram to the right details the redistribution of nuclei. The sexual reproductive structure in Basidiomycetes is a basidium, which typically produces 4 basidiospores. The rusts and smuts are 2 important groups of plant pathogenic Basidiomycetes. Many others are involved in wood decay, and Rhizoctonia is also a Basidiomycete. Many edible mushrooms are also Basidiomycetes, such as the button mushroom. They are also called gill fungi because the basidia are formed in gill-like structures in the fruiting bodies. The rusts are Basidiomycete fungi that attack a wide range of plant species, although most are host-specific. They are obligate parasites, and many are heteroecious, meaning at least 2 unrelated plant species are needed to complete their life cycle. Species that require only a single host are auticeous. Rusts get their name from the symptoms and signs of the urodiniospore stage, in which reddish-colored spores are visible in pustules that erupt through the leaf epidermis. 5 different spore types can be produced by rust fungi, although not all species produce all 5 of them. They are listed here in the order they are produced in rust disease of sweet corn, which will be described a little later, beginning with primary infection of the corn plant by aciospores. In this slide, you can see rust pustules on asparagus in the top image and sweet corn in the bottom image. Reddish urodiniospores have burst through the epidermis. Asparagus rust is caused by an autoecious species, Puccinia asparagi, while common rust of corn is caused by Puccinia sorghii, a heteroecious species. The following life cycle of the sweet corn rust fungus, Puccinia sorghii, illustrates the alternate hosts concept as well as the different types of spores. Let's start with the sweet corn plant, top center, infected with the fungus. The asexual or vegetative spores are urodiniospores, which are produced in pustules on living corn plants, as shown in the previous slide, and serve as a source of inoculum for other corn plants. That secondary cycle of spore production, release, and infection continues through the spring and summer. As the pustules mature, they produce teleospores, which are overwintering spores that germinate in spring to produce basidiospores. The basidiospores are spread by the wind, and those that land on the alternate host of corn, various oxalis species, can infect these plants and produce pycnospores, the sexual spores, and aciospores, which can infect corn. Aciospores, thus, are the primary inoculum for the corn plant. The smuts are also important plant pathogens. The example here is common smut of sweet corn caused by Eustigalomatus. The pathogen causes galls on almost any part of the plant. These galls are filled with mycelium and spores of the fungus. The name comes from the masses of black teleospores that emerge from the galls. The life cycle of common smut is much simpler than that of common rust of corn. Teleospores are the overwintering structures. When they germinate, they produce haploid sporidia. Sporidia of compatible mating types fuse and produce dikaryotic infection hyphae that infect the corn plant. We'll now move on to the Deuteromycetes, also called fungi imperfect and middlesporic fungi. The Deuteromycetes are the asexual or anamorph forms of Ascomycete and Basidiomycete fungi, which we touched on in our discussion on the one fungus, one name concept. In many cases, the sexual stage is known. For example, the sexual stage or teleomorph of Colototricum acutatum, an important pathogen of strawberries, peppers, and other fruit and vegetable crops, is Glomerella acutata. In others, such as the numerous alternaria species that attack fruits and vegetables, the teleomorph is unknown. In practice, the anamorph forms of many Ascomycete and some Basidiomycete fungi are the economically important forms, and the anamorph names are used in describing or otherwise talking about them. The morphology of the structures that produce conidia, as well as the morphology of the conidia themselves, is critical for the classification and identification of these fungi. These are the main types of structures producing conidia in the Deuteromycetes. Some species don't produce a structure at all, and conidia are produced on conidiophores on the plant surface. The four main structures producing conidia are Sporodokia, Cinema, Pycnidia, and Acervuli. Diagrams of each of these are shown in the next slides. In the upper left micrograph, free conidia, in this case in chains, are shown on a single conidiophore. A Sporodokium, top center, is a structure in which the conidiophores are grouped together, while a cinema is a more organized structure in which the conidiophores are fused. The pycnidium is a round or flask-shaped structure with an opening at the top that contains the conidia, which are called pycnidiospores. The pycnidiospores are formed on the conidiophores that line the inside of the pycnidium in a gelatinous mass that oozes through the opening when humidity is high or free moisture is present. The spores are dispersed by water splash. The Acervulus is an erumpent cushion-like fruiting body containing conidiophores and conidia. These conidia are also mainly dispersed by water splash. We'll move on now to the Oomycete, a smaller but very important group of plant pathogens. The Oomycetes are the largest group of economically important fungus-like pathogens. Historically, they are notable because one member of this group, Phytophthora infestans, shown in this micrograph, is the causal agent of the first plant disease to be worked on scientifically, called late blight. The loss of potato crops in Ireland due to late blight, combined with various socioeconomic and political factors in the mid-19th century, led to famine, starvation, and emigration on a large scale. The scientific work by Anton de Berry and others laid the foundation for the discipline of plant pathology. Because the Oomycetes produce zoospores that are motile in water, they are colloquially called water molds. The important members of this group are very aggressive plant pathogens. Key characteristics of Oomycetes are aseptate hyphae, asexual reproduction via zoospores with two flagella within sporangia, and sexual reproduction via differentiated gametes, the antheridia and oogonia, resulting in the production of thick-walled oospores. Oospores are resistant to adverse environmental conditions and can survive years in soil or other substrates. Heterothallic species require 2 different mating types for successful mating, while homothallic species self-fertilize. The 3 families within the order Peronosporales, most important as plant pathogens, are the Pithaceae, containing the genera Pythium, Phytophthora, and the recently named Phytopythium, the Peronosporiaceae, including the genera Peronospora, Pseudoperonospora, Plasmora, and Sclerospora, causing downy mildews, and the Albuginaceae, containing species of albugo that cause white rust. The characteristic reproductive structures of Pythium and Phytophthora species are the thick-walled oospores, shown in the upper left micrograph, lemon-shaped sporangia, usually with a papillum, which is shown in the right micrograph, of Phytophthora species, and the ovoid or branched cub-like sporangia of Pythium. Species in the Peronosporiacea cause downy mildews of many plants and are economically important in numerous fruit and vegetable crops. They are obligate parasites and cause typical symptoms on leaves that include yellowing and necrosis on the upper surface. The pathogens sporulate on the lower surface of diseased tissue via sporangiophores and sporangia. Downy mildew-causing genera are differentiated on the basis of the structure of their sporangiophores, as shown in the micrographs on this slide. The arrangement and number of sporangia on the sporangiophore, as well as the branching pattern of the sporangiophore, are key morphological features separating the genera. The last group are those that are important plant pathogens in the Albuginaceae family, mainly the genus Albugo. Albugo candida causes white rust in plants in the Brassica family. Although the name is white rust, the disease is completely different from rust caused by Pyxinia species. The name is related to the formation of white mycelium and sporangia on the undersides of infected leaves. Although the morphology of the sporangia and oospores is different than in the Pithaceae, the same types of structures are produced. The final group to consider in this course is the Plasmodiophoromycetes, and the only genus of economic importance in plants is Plasmodiophora, casual agent of clubroot, also of brassicas. This group is now considered to belong to the Phytomyxia. The life cycle and other characteristics of this genus will be covered in the Brassica Crops lecture. This is the end of the Introduction to Pathogens lectures. You will find instructions for taking the quiz in Carmen.